Yeah, thank you very much. It's been really enjoyable today listening to all the talks so far. So I hope I can meet the standard that's been set. So we'll talk about long-read microbiome analysis. Press the right button. Here's an outline of the talk. And we'll first of all just briefly talk about long reads. So we heard this morning from Nick about assembling short reads into MAGs. And there's a paradox here. You would think that, okay, if I have lots of short reads from my microbiome sample, I really want to assemble to get long sequences. So it would appear that it makes a lot of sense to assemble those short reads. But if you ever tried this, you know that it's always disappointing. What's called a MAG, a metagenome assembled genome, is not one or two contigs, it's hundreds of contigs of a very short length. So usually assembling short read microbiome samples gives you average contig lengths that are measured in the hundreds or the low thousands of base pairs, not really contigs by any serious standard. And so you might think, well, okay, so the paradox is you would think you should assemble short read microbiome samples, but actually it's not worth the effort. Whereas long reads, you might think, well, they're already 10 kilobases on average in length, say, from a MinION, so why assemble? The answer is because the longer your reads are, the much longer the assembled part's going to be. So long reads, you can often assemble into whole chromosomes. And so it turns out that when you do microbiome analysis using short read technologies, yeah, you can assemble and you can spend a lot of time binning, but you're never going to get chromosomes or anything close to chromosomes. Where long reads, you start off with long sequences, but you can get very long sequences. And you've probably seen pictures like this. This is just a random paper that I pulled that shows, what people look at when they've got short read data sets. You have a blizzard of tiny contigs, and you've got to figure out, which ones belong together. You can never be sure which ones really belong together. So I became interested in long read microbiome analysis as a visiting professor at NUS in Singapore, working with Rowan Williams, and then we're looking at wastewater treatment. And one of the early days when we started experimenting with a MinION, we did a run where we got 6 gig. When I say we, I should say Irena Bezarab, she did a run. She's the one who actually runs the sequencer that you can see down on the bottom right there. That's her sequencing station. So she got 6 gig out of a sample taken from an enrichment bioreactor, and 9 kilobase read length. And then we got very nice, using the unicycler pipeline back in those days, got very nice assembly that we then published. So this is the penultimate assembly graph before you actually cut it up into contigs. And you can see there's multiple closed chromosomes and so on. So this is what metagenomics should be like. You shouldn't be dealing with tiny, tiny pieces of sequence. You should be dealing with whole chromosomes and whole plasmids and so on. And that's what you can do more and more with long read sequencing technologies. So one pipeline that we've always worked on, but doesn't seem to have made it into the cubic connection, is Diamond plus Megan. So for long read analysis, what we like to do is take these sequences once they've been base called and then run an assembler. We used to use unicycler. More recently, we've been using Metafly to get fast stage sequences. Then we like to run Diamond against NCBI, but also against other databases. I'll get back to that in a moment, to find the coding sequences. Personally, I think that's the most interesting part of a metagenome. What genes do you have? Yeah, and so we look for the coding sequences, and then we do taxonomic and functional analysis based on those alignments. So the Diamond program has been upgraded by Benjamin Bulfing to specifically work well on long reads, and Meganizer also has a long read mode. You do this calculation on the server, and then you open your files in Megan. And you can either download the files onto your laptop or desktop, or there's also there's an HTML server that you can run on your server that serves files to Megan for you. And so more recently, and we're about to make this available, we've been using the ANOTREE protein database as an alternative to the NCBI NR database. Nick Youngblood mentioned this morning, that GTDB is probably a better taxonomy than the NCBI for purposes of metagenome analysis. And similarly, the corresponding protein database, the anode tree database, is probably a better database to align against than the NCBI-NR. So if you open up one of those files that you've produced with this pipeline, you see something like this. And this is actually, in Megan, we do support both taxonomies. So you can look at things using GTDB, but also you also have the NCBI view. So you have multiple views on the same data set. And basically you align once against a reference database, and then all the different analyses are built off of that one alignment run. So you get them more or less multiple analysis for free. Yeah, and that data that we looked at back in 2019 turned out that we had a number of high quality draft genomes, medium quality draft genomes, and some low quality draft genomes. And these terms, high, medium, and low quality, are really defined via a tool called CheckM, that you can give it a draft genome and ask it to evaluate the completeness and contamination. And people like to say that if you're over 90% complete and under, I think, 5% contaminated, then you're high quality. And then there's thresholds for medium quality and low quality. And you can see that we have nice completeness here and low contamination. And one thing, I don't know whether this is in your way as well. One thing, if you analyze long read data and you try to run something like CheckM or Procar on your long read data, you've probably been disappointed because it doesn't, those tools do not work out of the box. Nanopore sequencing puts frame shift errors into the sequence, or puts insertions and deletions into the sequences that will break frames when you're using a translated blast. So you have to use something like a frame shift aware alignment like Diamond provides. And so in Megan, you can actually, Megan provides a frame shift correction mechanism. So you put your long reads or Contigs into Megan, and you can export them in a frame shift corrected fashion. Then all the tools like CheckM and Procar work very nicely. You do get meaningful CheckM readouts, and you do get meaningful annotations of your long read assemblies. Without frame shift correction, then you don't, that doesn't work. Yeah, so here are those 7 chromosomes that we got back from that paper there. Okay, so long read metagenomics. If you want to explore this, we provided a resource. We've put together a resource to make it easy for people to look into long read microbiome analysis. And so if you fire up Megan and click on the file menu, then you'll see like on the 4th position, there's something open from server. And that allows you to open files that are being served from somewhere. And so we have multiple servers that we run in different projects with people, but we also have a public server that is pre-configured in Megan, so anybody can open this. And there we have a subdirectory long reads, and there we've put data from a number of studies that have recently been published. There was a paper 2019 with 22 human gut samples. There's some biogas wastewater samples, there's some seawater samples, groundwater samples, more wastewater samples, and some oral samples. Okay, these are all Oxford Nanopore. We're currently processing some PacBio, so we soon put some PacBio data there as well. And so you can just open these up on your laptop, and then you can explore these. You can have a look at the context length and what genes you can find and so on. And so I thought I would prepare something for this presentation that's less boring than just a bunch of slides. And so I opened up one data set from each of 6 different environments, and I would like to run a quiz here. Okay, so here you see 6 word clouds generated from 6 samples that I've opened. And one's from seawater, one's from groundwater, one's from human gut, one's from human oral, one's a wastewater, one's a biogas. And now what I was hoping is that we do this quiz. So I would like somebody to tell me which one of these 6 samples here is the seawater sample. So maybe just say it, turn your microphone on and say a number between 1 and 6. If you think you know which one of these samples is the seawater sample.
Number 5.
Number 5, okay. And why did you guess 5? I mean, that's what I would have guessed as well, as a non-biologist. Something when it's got plankton marina in it and stuff like that, that sounds very much like sea or Marinobacter, yeah. And so, yep, Daniel, thank you. That is correct. Okay, what about groundwater? Let me go over there and click on that. Okay, which one's got groundwater? Which is a groundwater sample? This is
4.
Okay.
No,
Why do you say 4?
2.
Okay. Okay, so 2 or 4. Anyone else support either 2 or 4? Just one more person so I can.
I think 2.
Turns out 4 is the correct answer. Okay, 2. I don't think it's going to be 2 because Candidatus accumiobacter, which is in 2 here. This is like the organism that does a heavy lifting in a wastewater. I shouldn't say what. Yeah, okay, I'm giving it away. Yeah, no, it is groundwater, and Legionella might also give it away. You'd hope that there's no Legionella in the human gut, the human oral. Yeah. Any case, yeah. And I don't know. So I should have checked this. I don't know. There's a fact that there's only a few genera here. Is that due to a small sample size, or is it due to groundwater being very poor in any case? Let's move on. Human gut, that's an easy one. In fact, because lots of people here study human gut. And we all know that there's going to be bacteroidetes and stuff like that in there.
There are several people in the chat saying 1.
Ah, okay, 1. Sorry, I can't see the chat. I should turn the chat on. Yeah, okay, 1. Yeah, thank you. Human gut. Human oral. Which one would that be?
3.
Yeah, 3. That's Streptococcus and Neisseria and those kind of things make that an obvious. And yeah, then the 2 last ones are wastewater biogas. At first, I couldn't really tell which is which, but then I guess, as I said, Candidatus accumiobacter accumulates phosphate in a phosphate reduction wastewater treatment plant. So that would give that away. So that's going to be wastewater and that's going to be biogas. So it's kind of fun that you can actually, just by looking at these word clouds, match things. So that's one of the things you can do. So you can just open up Mega and you can start looking at these different samples. As I said, we're also going to put some PacBio samples in there as well. Yeah. Okay. And if you ever want to use Diamond and Megan, we have recently published in current protocols a very detailed protocol, both for the short read and the long read case. Moving on to other topics. MAIRA is something that I've talked in some presentations about. So some of you might have already heard of this software that we developed in a DFG project. This was Benjamin Albrecht, who now works for CIRGAT here in the sequencing company here in Tübingen, and Jana Baci, who has now moved to Nadine Simat's group in biotechnology. And so this is a software that runs on a laptop and analyzes nanopore reads, and we envision this happening as the reads are being produced. So as reads are coming off the nanopore device and being base calls, you can have a look at what's in the sample. And we're particularly interested in what strains and particular types of genes. Yeah, so that's, and so it looks something like this. You fired up your nanopore sequencer and you're running base calling on something like the MinIT or on some graphics, the GPU, something like that. And as the sequences come out of the base calling, this program monitors the directory where they arrive, and then every now and then it updates its analysis. So something like that in this animation here. And so this does an analysis based on protein alignment. Again, we use that, seems to be our favorite idea. And because you're doing protein alignment now in real time or quite fast on a laptop, we use a 2-stage process where we first, as reads come in, to have a database where the proteins represent different genera. A genus represents, yeah, so we don't do a very detailed analysis. We just do a a lightweight analysis, trying to figure out which genera seem to be present in the sample. And once the score that we keep for each genus exceeds a certain threshold of 0.8, we then run the reads against a genus-specific database. And then, so you can see here, the first part of the analysis produces these blue genus level assignments, and then once we're confident that a certain genus is level, then all the reads are aligned also against the species databases. Yeah, so that's the basic idea. And the idea is to do this in a way that can keep up with the base calling. Yeah, and under the hood, Jana developed this idea of a protein synteny graph. It's a graph where each node represents a gene, and there are edges between genes if those genes appear adjacent to each other in some of the nanopore reads. So this gives you context. So especially if you're trying to figure out virulence factors and stuff like that, you need to know sometimes that certain sequences you're interested in are close to each other in the genome and not just somewhere in the microbiome. Okay, so you end up with a graph that looks like this or even way more complex. And that's what the analysis is based on under the hood. So we published that last year, and we're working hard to produce a major update in the next few weeks. Yeah, and so we had a lot of fun before the pandemic started in that Anjanen's lab. So here's Irina Bezarab, the colleague who I mentioned in Singapore, who was doing the nanopore sequencing. She came out to Tübingen to teach us how to do nanopore sequencing here in Laz's lab. And so we did run the MinION up here, and then we did use our software to monitor the content of to get an early readout of the content of a sample that we had from one of the bioreactors that Laz's group is studying. Yeah. More on the topic of bioreactors. Actually, this is going to be a really short talk, which is, I guess, not a bad thing. More time for discussion. I kept kicking stuff out until now. I probably kicked everything out, it looks like. Okay, bioreactor monitoring. So this is work that we're doing with Lars Anjanen and with Ruth Lay. So Lars and his group, what they like to do is to build bioreactors to grow microbes under controlled conditions. And they're trying to understand different processes. They're trying to optimize processes, figure out which pathways are doing what in many different contexts. Some of them more to do with biotechnology, but some of them also to do with what goes on in the human gut, using a bioreactor as a reduced complexity model for certain aspects of processing in the human gut. And so traditionally what people do when they run bioreactors is obviously they monitor all the experimental parameters, all the readouts of chemicals that go in, chemicals that go out, hydrogen that goes in, hydrogen that comes out, that kind of thing. And traditionally, what Lars Anjanen and others have done in the past is to collect biomass on some schedule, and then at some later date, send that all off for sequencing. Okay, so it was an after-the-fact analysis of what was going on in the bioreactor at the time. But obviously, with the MinION, where everybody can have their own little sequencer, that opens up the possibility that you could sequence on a regular basis and then figure out what's going on during the experiment, and maybe even design the experiment so that parameters are changed based on what is going on in terms of taxonomic and functional content. So the idea here is to monitor the community, the microbial community, using a MinION sequencer. And so that's a very nice project. So there's two PhD students, Ulrike Behind and Timo Lucas. And so Timo's in my group and he's doing the bioinformatics part, and Ulrike's in Laz's group and she's building the reactor. She goes to the glass blower and says, I need you to produce something that looks, has this shape and so on. I'm going to put an electrode here and there. So for us, it's quite exciting to work with people who do that kind of stuff. And what Timo's doing, now this is just a mock-up. This picture is grabbed from a paper by Lawrence David et al. This is another picture I grabbed off the web. And so what Timo is producing is a software that we might call MMonitor, which allows you to, once if you do sample from your microbiome on a regular basis, and you want to be able to just have a look at, what's going on in my bioreactor based on on the measurements of chemical properties or of the microbiome. So this is a tool that runs in a web browser. So there's a database on a server where the data is kept, and then anywhere, if you want to know what's going on what's been going on in my bioreactor in the last couple of weeks, you can open this up in your web browser and say, okay, I can see something bad happened here, and the microbiome has changed and has never come back to what it used to be. That kind of thing. So that's the first version of that is very close. And one more. So Diamond and Megan's been around since 2005, so that's not new. Diamond's been around since 2015, so that's not new. MAIRA's quite new. MMonitor is very new, and this is another new thing that I've been programming. A program that is called ALORA, Analysis of Long-Read Assemblies. And what I'm trying to build here is a tool that allows you that integrates multiple things that people like to do with long-read assemblies. Starting off with a Bandage type visualization of the different components of the assembly graph, where you can then open up one of these components and study it in more detail, but also showing the different contigs that you have, allowing you to sort them by different features, but also structure them. Say if you've got a taxonomic assignment on your contigs, you can show the contigs in a structured way. You can produce those typical plots that people like to look at, where you have depth of coverage versus GC, say, and then try and evaluate, and then have balloon size. I guess here we're using the size of the balloon to represent the length of the contigs, that kind of thing, together with, so that's some of the things that we put in there already. And I have a student who's doing a research project who's going to add a lightweight genome browser type view. If you're looking at your contigs in the context of an assembly graph, you might want to see, okay, are there any 16S's on any of these contigs, that kind of thing, or then transfer RNAs. So the basic idea here is to bring some of the analysis that we like to do forward to the assembly graph step before we actually decide what are going to be the final output of the assembly, what are going to be the individual contigs. We want to, So this will be an integrated viewer that runs off of a GFA file, where you can explore your long-read assemblies. Okay, so it's quite a short talk. Long-read, I think, for me at least, I like long-read metagenomics way more than I ever liked short-read metagenomics. I'm so glad that I don't have to deal with 100 million or a billion tiny reads that are assembled into tiny pieces. If you never tried this pipeline, you could try using assembly followed by Diamond followed by Megan, and then put that onto Megan's server, and then open those files. MAIRA, I described MAIRA, which is our laptop-based analysis while you wait tool. While you wait for the assembler the sequencer to finish, you could already start seeing what's in your sample. Then we've got the MMonitor for microbiome monitoring if you're running bioreactors and you want to do that in a systematic way. And then I just talked about ALORA. That will be a new tool for interactive analysis of long-read assemblies. And these are all the people I like to thank: people in my group, people in Singapore, Lars Angela and Ulrike, and Ruth, and these funding agencies, and you guys for listening to this talk. Thank you.